about that the aldoses and ketones having 5 or more carbon containing sugars mainly present in their cyclic form in the solution. They can either in the 6-membered pyranose or the 5-membered furanose rings. And then we also discussed about uh, these uh, these sugars mainly present in the chair conformation or the boat conformation, but the chair conformation is more stable than the boat conformation. The Fischer projection and the Haworth projection is only to understand the stereochemistry as well as the cyclization, but actually the sugars present only in the chair conformation. And then we also discussed about how to transform the Fischer to Haworth, Haworth to the chair conformation. We also said that the substituents that are present on the right side should be written down when we transform into the Haworth. Substituents that are present on the left side should be written up on when we transform into Haworth. And when the Haworth to the chair conformation, substituents that are down in the Haworth should also be written down in the chair conformation. Substituents that are up in the Haworth should also be written up in the chair conformation. But only thing is, we said that in chair conformation, we don't call it as a down or up. We say that either axial or the equatorial position. Then, so that means, for example, you can see here that here when we want to draw the structure of the D allose and D L allose, both Haworth and the chair conformation, all of you carefully see here when during cyclization, this is the carbon 1 and this is the carbon 2. Carbon 1 and carbon 5 are involved in the linkage. So in the same way, here also this is 1 and this is the 5. Both are involved in the linkage. Both are involved in the linkage. So now here, as I already said, that substituents that are present on the right side should be written down in the Haworth projection. Therefore, this is down and this is down and this is down and this is up, this is up, this is up when we transform into the Haworth. And this CH2OH group, it should point up in the chair, this one Haworth projection. And so now I will draw the Haworth projection for the D allose. We are, so this is the 6-membered pyranose structure, because only the pyranose form is more stable in case of aldoses. And so this is the carbon 1 and this is the carbon 2 and this is the carbon 3 and 4 and 5 and this is the CH2OH. This is the 6th carbon. So this is 1, 2, 3, 4, 5 and 6. So now, as I already said, that carbon 1, this is the carbon 1 and also the carbon 5. These two are already involved in the linkage. 5 and 2 are involved in, 1 and 5 are involved in the linkage. So therefore, so we forget about this one. Then we need to see the remaining. And here on second, OH is right. Therefore, we should write it down. And third, OH is right and it should be written down. And the fourth carbon, OH is right and it should be written down. And of course, if the OH is down means H is up here, OH is down means H is up, OH is down means H is up, and here H. And here during cyclization, again, another stereocenter is getting created here. And here, if the OH is down and H is up, this is the alpha D allose. And if the OH is, if in another way, if the OH is up and the H is down, then it is the beta D allose. So this is the way we need to transform this one. Coming to the same thing as I already said, that when you are writing the chair conformation, and again we need to draw the structure of the chair. And here, as I already said, that all the substituents can take up 2 orientations, either axial or the equatorial. So this is the carbon 1, this is the 2, this is the 3, and this is 4, and this is 5, and this is 6. This is 6. So this is axial bond and axial, 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 and axial, equatorial, 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 and equatorial. So now, when we are transforming from Haworth to the chair conformation, substituents that are present, the substituents that are present below the plane of the ring should be written down in the chair conformation as well. For example, we are writing here the alpha anomer. So OH is in axial position on the anomeric carbon. On the second carbon, OH is down, so it is in axial position. And on third, is OH is down, it is in axial position. And on fourth, OH is down, again it is in equatorial position. And of course, that's all. So now here again a H, here H, here H, here H. So this is the chair conformation of the D allos. And so this is for the allos. So coming to, this is for the D sugars. And coming to the L sugars, as I already said that there are some rules for writing the L sugar and the D sugar. They have it on the chair conformation. Since it is not a chemistry subject, it is only biochemistry. We need not go into the too much of chemistry details, especially the stereochemistry. So we should only know how to transform these structures. So now here, let me once again explain you the Haworth structure of the L allos and also the chair conformation. As I already said that when you transform this one, the things which are present on the down, and again here this is up, up, up. Only thing is in L sugars, the CH2OH group points down. That means here the CH2OH group, here H. You can see here. In case of D sugars, the CH2OH group, primary alcohol group, points up, whereas in case of L sugars, the CH2OH group into the ring. So that's, so when you look at any structure where the CH2OH group is into the ring, then we should understand that these are the D sugars. Remaining the same thing you can follow. If the substances that are present on the right, it should be written down, and the left means it is up, and the same way you can do this one. So, and of course, as we know, most naturally abundant sugars are the D sugars. So therefore, we don't bother about the L isomers. I just wanted to convey this message. That's why I'm just explaining this one. You need not bother about the L isomers. And then, and of course, as I already said that the sugars, as I said, that they can also form pyranose and the furanose structures, but the pyranose form is more stable, especially in aldoses. That's why we write only the pyranose, whereas in case of furanose, the link is between the aldehyde group and as well as the hydroxyl group on the fourth carbon. And of course, you can see here, this is the first, and this is the second, this is third, and the fourth, and this is the fifth, and this is sixth. The linkage is between C1 and the C4. So this is the furanose form of the this one. And now coming to this is same, as you can go through later. This is this will help you to understand how to transform the Fisher to Haworth to chair conformation in exam. And definitely you should practice all these structures for the examination point of view. Now, as I already said that you need not memorize any structures. Only thing is you need to remember the shortcut what I have given you, and with that, if you're able to write the structure of the D-glyceraldehyde for the D sugars, you will able to write the structure of any D series. In the same way, if you know the structure of the dihydroxyacetone, you will be able to do the structure of all ketoses as well. And then in yesterday's class, we discussed about the disaccharides. In the disaccharides, we initially we discussed about the maltose and the lactose and also the sucrose. and these are the three important disaccharides. And maltose is a polymer, it is a composed of two glucose units with alpha 1,4 glycosidic bond. Lactose with one galactose and one glucose with beta 1,4 glycosidic bond. And whereas the sucrose is composed of the glucose and the fructose with beta alpha 1 beta 2 glycosidic bond, and the sucrose is a non-reducing sugar because both the anomeric carbons are involved in the glycosidic bond formation. In addition to this, we have the isomaltose, where the two glucose residues are connected by the alpha 1,6 glycosidic bond, and also the silobiose, it is two glucose residues connected by beta 1,4 glycosidic bond. And then coming to and we also said that trehalose, that is a 1,1 glycosidic bond. And then coming to the polysaccharides. And in polysaccharides, we have already said at the beginning, polysaccharides mainly are classified into two types. One is homopolysaccharides, another one is the heteropolysaccharides. Homopolysaccharides have mainly either structural or the storage function. You can also see here they have either the structural role or the storage function, whereas the heteropolysaccharides they are mainly present in the extracellular component, in the sense they have the extracellular support, for example, peptidoglycan. In the same way, in animal tissues, these heteropolysaccharides mainly present in extracellular space, and that provides the structural support and holds the cells together and provides the protection, shape, and support to the cells and the tissues as well. So, and in general, when we say the polysaccharides, they are the long carbohydrate polymers, and they are also known as the glycans. We have already said that they are also known as the glycans, and they have molecular weight greater than 1,000 kilodaltons, and low molecular weight polymers are water soluble, whereas the bulky carbohydrate polymers are water insoluble. And they are classified into two types, homopolysaccharides and heteropolysaccharides. Homopolysaccharides are made up of with only one type of sugar residues, whereas the heteropolysaccharides are made up of with more than one type of sugar units. And homopolysaccharides are mainly either structural function or the storage function, whereas the heteropolysaccharides mainly provides the extracellular support, and they are also present in the extracellular space in the animal tissues and the cells, and they provide they hold the cells together and also provide protection and shape to the cells and tissues. And coming to we have the homopolysaccharides, we have cellulose and chitin, and they have the structural function, whereas the storage function remains the starch and the glycogen. All these are the homopolysaccharide. They are built with only one type of sugar residues. And whereas the heteropolysaccharide, important heteropolysaccharides, what we are going to discuss in this class is that glycosaminoglycans, GAGs. Glycosaminoglycans, they are mainly the hyaluronic acid, chondroitin sulfate, heparan sulfate, keratin sulfate, and dermatan sulfate, and so on. And all these GAGs play an important role in the animal and in the human body, or in other animals. And now let us go one by one. We will discuss about the first homopolysaccharides. First one is the starch. And starch, as you can see here, that the starch consists of two components, and one is amylose. Amylose, you can see here. Second one is the amylopectin. So that means starch is composed of two components. That is, one is amylose, and second one is amylopectin. Amylose is a linear polymer of glucose with alpha 1,4 glycosidic bond, whereas the amylopectin is a polymer of, again, glucose with alpha 1,4 as well as the alpha 1,6 glycosidic bonds. You can also see here, so amylose is a linear polymer, whereas the amylopectin is a branch polymer. And you can see here the alpha 1,6 glycosidic bond. This is the sixth carbon, and this is the first carbon. This is first carbon in the sense the ring is rotated here, and the branch points at the branch point alpha 1,6 glycosidic bond is present, whereas here this is the first carbon and this is the fourth carbon. This is alpha 1 for glycosidic linkage. So this is the structure of the starch. So that means starch is a linear polymer of glucose, and it has two components, amylose and the amylopectin. Amylose is a minor component, amylopectin is a major component. Amylose is a polymer of glucose with alpha 1 for glycosidic bonds. Amylopectin is a polymer of glucose with alpha 1 for as well as alpha 1,6 glycosidic bonds. And the alpha 1,6 glycosidic bond is present at the branch points, and these branch points occur present for every 20 to 30 residues once in the structure. And you can see this is the wherever you are seeing here, this is the branch point, and this is the branch point. That's where the alpha 1,6 glycosidic bond is present. And you can and of course this is you can see here, this is amylose and this amylopectin. It is a highly branched, it has many branch points, and it is you can also see here the amylose is a linear, amylopectin. So starch has both amylose as well as amylopectin component, and the major component is amylopectin, minor component is amylose. And we know that it is the storage polysaccharide, and because it also serves as reserve food material in the plants, and it is present in the many food grains, tubers, and so on. So this is about the starch. And coming to the glycogen. So glycogen, it is the glycogen. The glycogen is a storage polysaccharide in animals. We have already said at the beginning, and it is also a branch polymer of glucose. You can see here that it is also polymer of glucose with alpha 1,4 and also alpha 1,6 linkages. But the difference is that the branch point is present for every. You can see here, this is the branch point, this is the branch point, and this is the branch point, this is the branch point. The branch point is present for every 6 to 7 residues, residues one in the main chain. And that means the structure of the glycogen is similar to the structure of the amylopectin, but only difference is that this glycogen has more branch points compared to the amylopectin. And this glycogen is mainly present in the liver and the muscle, and the human body stores this excess of sugars in the form of the glycogen. And when the body demands energy, the glycogen would be further converted into the glucose, and the energy would be, and that glucose enters into the glycolysis and the TCA cycle, and in order to produce the high energy molecule ATP to run the metabolic processes. So that means the excess of glucose is stored in the form of the glycogen. In the sense, if the glucose, the excess of glucose, if it is stored in its monomeric form, that is in the form of the glucose, the cell osmolarity would drastically raise. As a result, the cell death takes place. That's why the body stores excess of glucose in the form of the glycogen in the human body, and mainly in the liver and the muscle. So this is about the glycogen. And coming to the third homopolysaccharide, that is the cellulose. So cellulose, we know that it is present in the cell wall of the plant, and it is a linear polysaccharide of glucose. Again, you can see here, these are the all glucose residues, and you can notice here that this is the beta 1,4,1,4 glycosidic bond. This is the first carbon and this is the fourth carbon. So that means the cellulose is a polymer of glucose linked by beta 1,4 glycosidic bonds, and it is present in the plants in the cell wall. And you can see here that if you see the structure of the cell wall, and the cell wall has several the cell wall has many the cellulose microfibrils. Each cellulose microfibril has several cellulose molecules. All these cellulose linear chains are connected to each other by the interchain hydrogen bonding, and this interchain hydrogen bonding gives the rigidity to the cellulose that gives rigidity to the plant cell wall. And most of the animals cannot use these cellulose as the fuel source because they lack the enzyme to hydrolyze the beta 1,4 glycosidic bonds. But whereas the termites can readily digest the cellulose because the termites harbor certain bacteria in their intestine, and that symbiotic bacteria secrete enzymes that digest the beta linkage, beta 1,4 linkages here. That's why the termites and also the ruminant animals such as cattle, deer, giraffe, camel can also digest the cellulose because the bacteria that live in the rumen also secrete a cellulose enzyme. But and as I already said that the extensive the linear chains are held together tightly by the hydrogen bonding, and that gives the that this hydrogen bonding stabilizes the structure and also gives the rigidity to the cellulose. And this is about the cellulose. Now coming to the dextran. Dextran, you can see here that it is also polymer of glucose, and you can notice here glucose with alpha 1,6 glycosidic bond. You can see here alpha 1,6 glycosidic bond. So that means it is a polymer of glucose with alpha 1,6 linkages. But the branch points, you can see here, branch points are present at with alpha 1,3 or alpha 1,4. So that means it is a polymer of glucose with alpha 1,6 linkages with a branching either at alpha 1,3 branch points or with alpha 1,4 branch points. And this dextran and most of the bacterial and yeast polysaccharides, bacteria and yeast polysaccharide also have this dextran. And the dental plaques, what you are seeing here is dental plaques that are formed by the bacteria are growing on the surface of the teeth is also rich in dextran. That means these dental plaques are because of the dextran. And the synthetic dextran, what we use the Cepadex. So this is the synthetic dextran, and the Cepadex that is also used as a stationary phase material in for the purification of molecules in the gel filtration chromatography. So in gel filtration chromatography, the synthetic dextran Cepadex is used as a stationary phase material for the as a stationary phase material in the gel filtration chromatography. So this is a dextran. It is a polymer of glucose with alpha 1,6 linkages with branch points at alpha 1,3 or the alpha in some cases alpha 1,4. And coming to the chitin. So chitin, it is a chitin, as we already said, that it is present in the hard exoskeleton of many arthropods, insects, lobsters, and crabs. And this chitin is a polymer of, you can see here, this is the N-acetyl glucosamine. It is a polymer of N-acetyl glucosamine with beta 1,4 linkage. This is the first carbon and this is the fourth carbon, and the linkage is beta linkage, beta 1,4 linkage. So this is the structure of the, it is also linear polymer of glucose with beta 1,4 glycosidic bonds. Linear polymer of N-acetyl glucosamine, not the glucose, with the beta 1,4 glycosidic bonds, and it is mainly present in the exoskeletons of many arthropods. And these are about the, and in addition to this, we also have, you can see here, this is the structure of the N-acetyl glucosamine, and you can see here the linkage is a beta 1,4 linkage. And there are, I think, many more, but we are discussing only few. Next, the homopolysaccharide is the inulin. So inulin you can see here it is a polymer of fructose. So these are all fructose residues. It's a polymer of fructose with the terminal glucose. You can see here, this is the glucose with a terminal glucose and terminal glucose. In the sense, these inulins are the storage form of carbohydrates in the tubers, especially the dahlia, artichoke, onions, garlic, bananas, and asparagus, chicory, and so on. Since it is a polymer of fructose, and the inulins are also called as the fructans. The inulins are also called as the fructans, and the linkage is here is the beta 2,1 linkage, and the glucose is present at the terminal point with again the beta 2,1 linkages. So this is about the inulin, and you can also see here this is fructose and this is sucrose, and this is the several fructose units are connected by beta 2,1 linkages and with a terminal glucose. So that's why, since it is a polymer of the fructose, that's why the inulins are also known as the fructans. So these are all polysaccharides what we have discussed so far, that is the starch, glycogen, dextran, inulin, cellulose, and chitin. So now we already said that all these sugar, all these polysaccharides are built with only one type of sugar monomeric units. And coming to the heteropolysaccharides, we have mainly the glycans. In addition to this, we have another two more we will discuss here, that is the peptidoglycan. So we have already said the peptidoglycan is present outside the plasma membrane, and the peptidoglycan is multi-layered in case of gram positive, and it is a very the thickness of the peptidoglycan is very less in case of the gram negative organism, and this peptidoglycan gives the protection to the organism to sustain the internal osmotic pressure that is developed. And if you see the chemistry of this peptidoglycan, and this peptidoglycan is a polymer of N-acetyl glucosamine and N-acetyl glucosamine and N-acetyl meramic acid, again NAG, again NAM, and so on. So that means it is a polymer of NAG, NAM, NAG, and NAM, and with beta 1,4 linkages, beta 1,4 linkages. And you can see here that this is the N-acetyl glucosamine, N-acetyl meramic acid, N-acetyl glucosamine, N-acetyl meramic acid. They are connected by beta 1,4 linkages, and this, since the peptidoglycan is a multi-layered and one NAG and NAM layer is connected to another NAG and NAM layer through a short peptide. You can also notice that one here. You can notice here that one NAG and NAM layer is connected to the another NAG and NAM layer through a short peptide, and this this peptide bond is catalyzed by the transpeptidase enzyme. And what exactly happens here is that if the one NAG and NAM layer is not cross-linked with another NAG and NAM layer with a short peptide, the peptidoglycan becomes very loose. So that's why this cross-linking is very important for the peptidoglycan. So the enzyme lysozyme, we know the enzyme lysozyme they kills the bacteria by hydrolyzing the beta 1,4 glycosidic bond here, beta 1,4 glycosidic bond between the NAM and NAG, but not between the NAG and NAM. It is because it is a highly specific, enzymes are highly specific. So these lysozyme hydrolyzes the peptidoglycan by hydrolyzing the beta 1,4 glycosidic bond between NAG and between NAM and NAG, but not between the NAG and the NAM. And in the same way, the penicillin also inhibits the growth of the bacteria by preventing the synthesis of this cross-linking. As I already said, that one NAG and NAM layer is cross-linked with another NAG and NAM layer through a short peptide, and this cross-linking is catalyzed by the transpeptidase enzyme. So penicillin inhibits this transpeptidase enzyme. As a result, the NAG and NAM layer synthesis takes place, but without cross-linking. As a result, the peptidoglycan becomes too loose, and as a result, the organism is not able to tolerate the internal osmotic pressure. So that means the penicillin, the lysozyme hydrolyzes the glycosidic bond between NAM and NAG, whereas the penicillin kills the bacteria by preventing the synthesis of the cross-linking by inhibiting the transpeptidase enzyme that is essential for the cross-linking of one NAG and NAM layer with the another NAG and NAM layer. So this is about the peptidoglycan. Coming to the agar. So this we know this we use the agar and agarose. These are the marine mainly produced by the marine red algae, and this is this agar is a polymer of the agar and agarose. These are the agarose and agaro. The agar has the agarose and agaropectin, and agarose is a polymer of the galactose and 3,6-anhydro L-galactose. So this is galactose 3,6-anhydro L-galactose with beta 1,4 and alpha 1,3 glycosidic linkages, and this agarose is also used as a medium for the electrophoresis in the for the substitution of the DNA and also the high molecular weight proteins. And the saccharose is a processed form of the agarose, mainly used in the electrophoresis and the electrophoresis. And this is about this one we have already said that inulin. So these are about the different types of the polysaccharides, mainly homo and hetero, and we have important class of heteropolysaccharides. So those are known as the glycosaminoglycans. These glycosaminoglycans, it is this topic is a very important. These glycosaminoglycans are also known as the complex sugars, and they are also known as the mucopolysaccharides, and because they are found in the mucus secretions first, and they have the lubricating nature. And this topic is very important in the examination point of view. And these GAG chains, glycosaminoglycan GAGs, are mainly present in the, as I already said, that the heteropolysaccharides mainly present in the space between the cells. So that means the extracellular space so is filled up with many GAGs, many glycosaminoglycans, and these glycosaminoglycans have the provides the lubricating property, and they are present in the animal cell surface and also the that is in the extracellular membrane. And these glycosaminoglycans are also known to bind to various proteins such as chemokines, cytokines, growth factors, morphogens, enzymes, and cell adhesion molecule. And if you see here, this is the extracellular matrix. That means all these glycosaminoglycans present in the extracellular matrix, they provide this one. And now coming to the major glycosaminoglycans that are present in in the human body are classified into two categories. One is sulfated, and the second one is non-sulfated glycosaminoglycans. And the sulfated glycosaminoglycans, and you can and before going to this classification, let me say a few words about these glycosaminoglycans are also the very long linear polymers, and they are the polymers of repeating disaccharide units. This point you need to remember. They are the polymers of repeating disaccharide units, and these glycosaminoglycans are can hold the large volume of water, and you can also see here. That's why they can provide the lubricating in the and also the load bearing capacity to the cartilage, because then they are present in the extracellular matrix. And when you can also see here, so that means they can hold large volumes of water. When there is a compression, the water that is surrounded by the GAG chains squeeze out, and occupy a small volume. When the compression is removed again, the glycosaminoglycan retains their original hydrated volume, and this property makes provides the viscoelastic and the load bearing capacity to the cartilage, because these glycosaminoglycans are also present in the cartilage. And these glycosaminoglycans are classified into two types. One is non-sulfated glycosaminoglycans. One is non-sulfated glycosaminoglycans. Second one is the non-sulfated GAGs. Second one is the sulfated GAGs. In a non-sulfated GAGs, we have only one glycosaminoglycan, which is non-sulfated, that is the hyaluronic acid. Whereas under sulfated glycosaminoglycans, we have chondroitin sulfate, and the dermaton sulfate, and the keratin sulfate, and of course it is keratin sulfate, and also the heparan sulfate. So all these are sulfated glycosaminoglycans. So that means glycosaminoglycans are negatively charged polysaccharides, and they are classified into two types. One is non-sulfated GAGs. Second one is sulfated GAGs. In non-sulfated GAGs, hyaluronic acid is a non-sulfated GAG, whereas under sulfated GAGs we have chondroitin sulfate, dermaton sulfate, keratin sulfate, and heparan sulfate are the heparin. All these are the this one. So coming to its structure, so all these glycosaminoglycans, if you see the basic structure of the GAGs, structure of the glycosaminoglycans, as I already said that glycosaminoglycans, they are composed of several repeating disaccharide units. Several repeating disaccharide units. So that means they are the polymers composed of several repeating disaccharide units, and each disaccharide repeat has one uronic acid, one amino sugar. So that means if you take any glycosaminoglycan, whether it is a hyaluronic acid, chondroitin sulfate, dermaton sulfate, keratin sulfate, or heparan sulfate, in all they are the polymers of repeating disaccharide units. That means this disaccharide unit is present several times. So that means they are the polymers made up of several disaccharide repeat, and each disaccharide repeat has one uronic acid, one amino sugar, and the commonly found uronic acid is D-glucuronic acid. This is the three-letter abbreviation for the glucuronic acid, and L-iduronic acid. Glucuronic acid and L-iduronic acid. These are the commonly found uronic acids in the disaccharide repeat. Coming to the amino sugars, the commonly present the amino sugar in the disaccharide repeat of the GAGs chains is that the amino sugars are the glucosamine. Second one is the galactosamine. These are the amino sugars that are present in the GAG chains. That means each disaccharide repeat has one uronic acid, one amino sugar. The commonly found uronic acid is the glucuronic acid or L-iduronic acid, and the amino sugar is glucosamine and or galactosamine. So this is about this one. So now coming to the we will discuss one by one in detail hyaluronic acid, chondroitin sulfate, dermaton sulfate, and keratin sulfate in the afternoon class.